Hello everyone, my name is Madison Lloyd and today I'm going to be presenting a research paper that covers the topic of microbiome resilience to antibiotic disruption. This paper was written in 2024 and the title of it is Commensal Antimicrobial Resistance Mediates Microbiome Resilience to Antibiotic Disruption. There were two main questions that the researchers sought to answer. They first wanted to discover how the gut microbiome adapts to long-term antibiotic treatment, and then to take this research one step further, they also wanted to see how this potential adaptation of the microbiome influences known inflammatory responses to infection. There are three things that I'm going to discuss pretty quickly as a background section before we get into the research. One being antimicrobial resistance, then we have the second one as GI microbiome disruption, and third, I'm going to discuss the multi-drug resistant tuberculosis treatment that is pretty extensively covered in this research. Although antibiotics have obvious health benefits when it comes to clearing and preventing infections, they are also capable of majorly disrupting the microbiome and causing antimicrobial resistance. Antimicrobial resistance is when microorganisms such as bacteria and viruses evolve to become resistant to antimicrobial drugs such as antibiotics. Antimicrobial resistance has been declared by the CDC to be a global health threat because it makes infections not only harder to treat, but it also increases the risk of diseases spreading. It is pretty well known that antibiotic exposure, specifically long-term exposure, can severely disrupt the gut microbiome. In this study, researchers examined the gut microbiome from patients undergoing something called multi-drug resistant tuberculosis treatment. They chose to concentrate on this treatment because it is representative of one of the longest antimicrobial exposure in humans, with a 20-month-long treatment cycle involving multiple antibiotics. The researchers from this article have also previously performed similar research on the gut microbiome after something called drug-sensitive tuberculosis treatment, which is standard tuberculosis treatment. Multi-drug resistant tuberculosis treatment involves a longer treatment course with 5 broader spectrum antibiotics, and the drug-sensitive tuberculosis treatment that they had previously researched is a significantly shorter treatment cycle involving 4 antibiotics that are not as broad spectrum as the 5 from the multi-drug resistant treatment. The researchers were able to discover two things. One, that the microbiome does in fact prove resilient, and it does recover from long-term antibiotic treatment. It was proven once again that long-term antibiotic treatment left the gut microbiome in an altered state during treatment, yet upon treatment cessation, the microbiome was repopulated. The source of this microbiome resilience was shown to be due to commensal antimicrobial resistant bacteria repopulating the microbiome. Commensal bacteria are microorganisms that are naturally present in a host that do not cause harm. They have actually proven to be pretty beneficial for various processes involved in digestion, metabolism, immune function, and brain function. The second thing that researchers were able to prove during this study was that inflammation clearance is based on not only the clearance of the tuberculosis infection or pathogen clearance, it is also based on the dynamics of the microbiome. This study aimed to find the antibiotic effects on the gut microbiome by examining multiple stool collections from 24 patients at different time intervals undergoing the multi-drug resistant tuberculosis treatment. The enrollment criteria was that these patients had to be diagnosed with tuberculosis based on the symptoms they were presenting, a chest x-ray that was consistent with tuberculosis presence, a positive molecular test, and the previous failure of the drug-sensitive or standard tuberculosis treatment. Researchers collected fecal samples at different intervals, including day 0 to show pretreatment levels, and then again at 2 weeks, 1 month, 2 months, and 6 months into treatment, as well as at treatment completion. They also measured the tuberculosis load in sputum samples at these same intervals to measure the disease resolution in the 24 patients. Antibiotics used to treat these patients included betaquiline, linezolid, levofloxacin, clofazimine, and this one's hard to say, so excuse me, but pyrazinamide. At the 6-month mark, betaquiline was stopped, and at the 12-month mark, the nelezeloid was stopped, with all of the other antibiotics I listed being continued for the entire 20 to 24-month duration of treatment. The levels of the tuberculosis bacterial load were reduced by various rates, with some patients achieving culture negativity at 1 month into treatment and all others achieving culture negativity at 6 months or by treatment completion. This is the first image that I'm going to present to you guys. The figure on the left is for species abundance, and the figure on the right is for metabolic pathway abundance. So researchers determined the changes in the GI microbiome during treatment using metagenomic sequencing of the stool samples. They looked at the abundances of both the bacterial species and the metabolic pathways that were present, and they were able to conclude that the multi-drug resistant tuberculosis treatment disrupts both the taxonomic and functional microbiome composition. For both of these images, there are box plots at the top and at the right of the scatter plots, and these show the distribution of samples at various time points. The main scatter plot for each of these species abundance and pathway abundance have different colors which indicate the different sample times of the various species or pathways present. The researchers were able to equate 23.6% variation in species abundance and 40.1% metabolic pathway abundances to changes in the microbiome caused by the multi-drug resistant tuberculosis treatment, as you can see in these two figures. Researchers were able to compare the abundance of microbial species and pathways to pre-treatment presence and their prior findings of species abundance in their research on the standard drug-sensitive tuberculosis treatment. The researchers discovered that multi-drug resistant tuberculosis treatment caused a depletion in the microbiome of the GI tract in the first 6 months of treatment. This figure on the right shows the taxonomic abundance during treatment, and if you can see where I highlighted yellow, those are some of the species that were discussed in this article that were almost, if not completely, depleted by the treatment. These findings align with what the researchers had already discovered when investigating microbiome depletion in the standard tuberculosis treatment, but this research showed more taxa than what was depleted by the standard treatment. Different from their results regarding standard treatment, the multi-drug resistant treatment showed to cause an expansion in certain pathogens such as Klebsiella pneumoniae, Klebsiella varicola, and E. coli. Majority of species lost within the treatment returned to baseline by 6 months and were almost completely restored by the end of treatment, which proved the resilience of the microbiome. In addition, 33 metabolic pathways were also depressed during the multi-drug resistant tuberculosis treatment, but between 2 weeks and 6 months of treatment, several pathways known to be associated with inflammatory conditions became present. One pathway in specific was a known signature of inflammatory bowel disease. Similar with the microbial species returning to abundance by treatment cessation, the pathways did as well. In summary, both species and pathway abundance are significantly depleted upon beginning the multi-drug resistant tuberculosis treatment, and which is accompanied by the enhancement of inflammatory opportunistic pathogen presence. Researchers were able to expand their prior research once again involving the inflammatory state that accompanies a tuberculosis infection, concluding that inflammation is resolved by both the depletion of the pathogen and the microbiome alteration. Inflammation is reduced as the pathogen level is reduced, which is a result of the antibiotic treatment. Researchers discovered that in addition to that, the alterations in the microbiome resulting in higher levels of Clostridia, which is a commensal bacteria, also reduced the presence of inflammation. The picture on your screen is just a cool microscopic picture of Clostridia. The researchers observed a pretty significant reduction in the expression of signaling pathways that were commonly associated with inflammation during treatment. They used an RNA sequencing method called blood transcriptome sequencing, which shows the RNA transcripts in a blood sample to see this reduction. The researchers then used statistical analysis called Mixed Effects Random Forest Regression Model. This model combines two different tools in statistics, the first one being something called the Mixed Effects Model, and the second one being the Random Forest Regression Model. By using this combination, they were able to see nonlinear relationships between the inflammatory pathways that were present while undergoing treatment, and also considering the variability present within the cohort of participants. This allowed the researchers to have a more accurate prediction of pathway presence and tuberculosis signatures. They then used another model in statistics called the Univariate Linear Mixed Effects Model to establish a positive and negative relationship between the tuberculosis signatures and the pathways present. The researchers were able to amplify the results of their prior research due to the multi-drug resistant tuberculosis treatment being a more severe and longer duration. The results they found in this study compared to their last one were more prominent changes in the presence or absence of pathways and species. So they were able to really see the microbiome's role in inflammation clearance. It is well known that long-term broad-spectrum antibiotic treatment causes increased antibiotic-resistant genes in the gut microbiome. The researchers in this study quantified multi-drug resistant tuberculosis treatment effects specifically on the rhizostome of the gut using something called shotgun metagenomics. They quantified these effects based on duration, magnitude, and resistance category. Shotgun metagenomics is a sequencing technique that is used to study the diversity present, and the nice thing about it is that you are not required to culture a microbe beforehand. This approach allows for a more comprehensive look at what genetic diversity is present because it doesn't just look at a specific microbe or a specific region, compared to some sequencing techniques that we saw in Module 1, like 16S, where it's pretty specific to a region. Since shotgun metagenomic sequencing is so much more comprehensive, it allowed for the researchers to not just see the identification of what was present, but it also gives a really good insight into the function as well. This study presented research supporting the significant presence of something called antibiotic resistant genes during the multi-drug resistant tuberculosis treatment. After 2 weeks of the multi-drug resistant treatment, 97 antibiotic resistant genes were present across the entire group of participants in this study. 86 of the 97 antibiotic resistant genes were still present at the 2-month mark into treatment, and 75 of those 97 were still present at the 6-month mark into treatment. Most of the antibiotic resistant genes did return to baseline after treatment had ended. The image on the screen right now, there's 5 pie charts, one for 2 weeks, one for 1 month, one for 2 months, for 6 months, and then TC stands for treatment completion. And then the bottom right, you can see the different categories of antibiotics that are present in these pie charts. And these pie charts are just showing the percentage of resistant genes that are present throughout those categories of antibiotics from the entire study, so all 24 participants. You can see how many different classes of antibiotics had antibiotic resistant genes present between the different intervals. And I thought it just really showed how there was such a crazy change. When you look at 2 weeks, 1 month, 2 months, and 6 months, you see a bunch of different colors, a bunch of different percentages, and then at treatment completion, there's just 3 divisions in the pie chart. So pretty cool to see visually. It has been proven in other research that after short-term antibiotic treatment, the gut microbiome stays in a state of dysbiosis, and it will recover but may never return to baseline. Microbiome resilience observed specifically in this study was proven to be caused by returning members of Clostridialis class of bacteria. One hypothesis presented in this section of the paper was that the returning bacteria may be in the same class as what was present before, but could possibly be different strains. These researchers looked for very finely detailed differences in metagenomes for different taxa between samples taken at day 0 or pre-treatment, and then samples taken at 2 weeks, 1 month, 2 months, 6 months, and at treatment completion. They used something called metagenomic intraspecies diversity as a tool to analyze the single nucleotide variants. They specifically looked at Klebsiella and E. coli and found that there were differences in the nucleotides between the strains present at the pre-treatment mark and the post-treatment mark. Then they analyzed commensals and found that there was a higher diversity in the single nucleotides at treatment completion, proving that those bacteria had mutated as well. Researchers then used something called the N-strain pipeline, which is a bioinformatic tool to further examine the mutations that were highlighted through the use of the metagenomic intraspecies diversity analysis. The use of N-strain allowed for a more detailed analysis in those single nucleotide variants. The researchers were able to identify even more mutations that were not shown through the metagenomic intraspecies diversity analysis, just further proving their point that those bacteria at post-treatment had mutated because of the antibiotics. This proved that the dominating pathobients did in fact undergo the resistance mutations, and these mutations are proof of microbiome resilience after long-term antibiotic treatment. This is the last little bit of information that I'm going to present from the research article. Personally, I thought this was the most fascinating part of the article, so I hope you feel the same. After proving that the microbiome is resilient to antibiotic treatment with the help of commensals, these researchers wanted to see if this resilient microbiome was functionally resistant to antibiotic disruption. So to do this, they took mice and they colonized them by fecal matter transplant. They had two different groups. They had a healthy community control group, and then they also had the other half that was colonized by transplants from those multi-drug resistant tuberculosis patients. After the fecal matter transplant, some of the mice were treated with beta-aquiline for just 1 week. And if you remember from the beginning of this presentation, when I discussed the 5 different antibiotics that are used in the multi-drug resistant course of treatment, the beta-aquiline was the one that was stopped the soonest. It was only given to the patients for 6 months, and then they didn't have it for the rest of that potentially 20-month-long treatment. After the transplant, but before the treatment of beta-aquiline, the mice that had received the transplant from the multi-drug resistant tuberculosis treated patients showed an abundance of 58 different species when they were compared to the healthy community control group of mice. After the beta-aquiline treatment, the mice that were in the multi-drug resistant tuberculosis treated group showed a closer proximity to their pre-treatment levels compared to the healthy community control group of mice, which did not show that much of a comparison. This confirmed that the multi-drug resistant tuberculosis treated individuals have a microbiome that is resilient to antibiotic disruption of antibiotics that were previously administered during treatment. Researchers were able to prove that the antibiotic resistance mutations that are present in commensal bacteria after antibiotic treatment help to promote microbiome resilience, and that these adaptations in the microbiome do help with inflammation clearance. It is important for research to continue to progress in understanding the behavior of the gut microbiome after antibiotic treatment due to antimicrobial resistance being such a pressing public health concern. So I have some questions to discuss. I have 5. The first one is, based on the information presented in this research, do you think alterations to pathobient genomes make them more or less likely to become pathogenic, and why? My second question is, what could be some possible negative effects of the commensal bacteria responsible for repopulation of the gut microbiome being resistant to antibiotics? The third question is there another sequencing technique that could have been more beneficial to use instead of shotgun sequencing, and why? The fourth question is, how do you think a deeper understanding of microbiome resilience could affect decisions involving antibiotic treatment? The fifth question is my favorite, and I'm actually really curious to hear everyone's feedback on this one, but I do have to give a little bit of a background. In the article, because these researchers had previously done research on the drug-sensitive tuberculosis treatment, and now they were looking at the multi-drug resistant treatment, they explained that when a patient fails the drug-sensitive tuberculosis treatment and then is put on that multi-drug resistant treatment, that multi-drug resistant treatment is tailored towards whichever antibiotic they showed resistance to, so they would exclude that one from their new plan of treatment. And it didn't ever specifically say in this research if all 24 patients had shown the same resistance to whatever they received in the drug sensitive treatment or anything like that. So I don't know if this was like a flaw in the research. Potentially it could have been that maybe they shouldn't have all been treated with the same 5 antibiotics, or they should have clarified that all 24 patients did show resistance to the same class of antibiotics in that drug sensitive treatment, and we're now being treated with these 5 based on that resistance shown. So the question is, MDR TB treatment is usually tailored towards the patient showing resistance to one of the 4 antibiotics used in standard TB treatment. What effect do you think this might have on the study based on the fact that all 24 patients received the same exact 5 antibiotics without considering which one had caused them to fail the standard tuberculosis treatment they received? So thank you guys for listening. I do have a page of sources if you're curious to see more. I hope you guys enjoyed this presentation. Thank you.